Introduction. Hello students. Today, we will be diving into the fascinating world of serine metabolism. Serine is a vital amino acid that plays a crucial role in various biochemical processes within the human body. In this lecture, we will explore the biosynthetic pathway of serine, the significance of an enzyme called serine hydroxymethyltransferase, SHMT, and its link to 1-carbon metabolism. We will also discuss the role of serine in neurotransmitter synthesis, its interplay with glucose metabolism, and the diseases associated with serine deficiency. Furthermore, we will shed light on the implications of serine metabolism in cancer cells and its regulation. Additionally, we will explore the potential of serine as a diagnostic or prognostic biomarker, the effects of serine supplementation on cellular metabolism, its role in cellular antioxidant defense systems, its connection with mitochondrial function, and finally, we will conclude our lecture by summarizing the key takeaways. So, let's begin our journey into the intricate world of serine metabolism. To understand serine metabolism, we first need to explore its biosynthetic pathway. Serine synthesis predominantly occurs through the enzyme-catalyzed conversion of 3-phosphoglycerate, 3-PG, an intermediate of glycolysis. The enzyme responsible for this conversion is called phosphoserine aminotransferase, PSAT. PSAT transfers the amino group from glutamate to 3-PG, forming phosphoserine. Next, phosphoserine phosphatase dephosphorylates phosphoserine, yielding serine. This dephosphorylation is a critical step in the pathway, as it ensures the availability of free serine for various downstream processes. One of the key enzymes involved in serine metabolism is serine hydroxymethyltransferase, SHMT. This enzyme plays a crucial role in interconnecting serine metabolism with 1-carbon metabolism. SHMT catalyzes the reversible conversion of serine to glycine, while simultaneously generating a 1-carbon unit that can be utilized for various biosynthetic reactions. This 1-carbon unit is carried by a molecule called tetrahydrofolate, THF, which serves as a coenzyme for SHMT. The interplay between serine and 1-carbon metabolism is vital for processes such as nucleotide synthesis, DNA methylation, and histone modification. 1-carbon metabolism encompasses a network of biochemical reactions involved in the transfer of 1-carbon units within cells. This intricate network relies on various enzymes and cofactors, including THF, vitamin B12, and samarium. Serine plays a critical role in 1-carbon metabolism, as it serves as a major source of 1-carbon units through the action of SHMT. These 1-carbon units can be used for the synthesis of nucleotides, amino acids, and lipids, thus contributing to essential cellular processes, including cell division and growth. Serine is not only essential for protein synthesis but also plays a significant role in neurotransmitter synthesis. It serves as a precursor for the neurotransmitters glycine and serotonins. Glycine is a neurotransmitter involved in inhibitory signaling within the central nervous system, while serotonin is a neurotransmitter that regulates mood, appetite, and sleep. The availability of serine is crucial for the efficient production of these neurotransmitters, ensuring proper neuronal function and communication. While glucose is the primary fuel source for cells, recent studies have shed light on the interplay between serine and glucose metabolism. Serine synthesis is closely linked to glucose metabolism through the enzyme 3-phosphoglycerate dehydrogenase, phosphoglycerate dehydrogenase. Phosphoglycerate dehydrogenase catalyzes the first step in serine biosynthesis, converting 3-phosphoglycerate derived from glucose into 3-phosphohydroxypyruvate. This interconnection between serine and glucose metabolism highlights the intricate metabolic regulatory networks that ensure a constant supply of serine for various physiological processes. Serine deficiency diseases are rare genetic disorders characterized by impaired serine synthesis or utilization. Patients suffering from these disorders exhibit a wide range of neurological manifestations, including severe intellectual disability, seizures, microcephaly, and developmental delay. These conditions underscore the importance of serine in proper brain development and function and highlight the critical role of serine metabolism in neurological health. Cancer cells often exhibit altered metabolic pathways to meet their energy and biomass requirements. Serine metabolism has been identified as a critical player in cancer cell growth and proliferation. Cancer cells hijack serine metabolism to fuel their increased demand for nucleotide synthesis, redox balance, and antioxidant defense. Moreover, the dysregulation of serine metabolism in cancer cells offers potential therapeutic targets for the development of anti-cancer drugs. Like any metabolic pathway, serine metabolism is tightly regulated to maintain cellular homeostasis. Several signaling pathways and transcription factors contribute to the regulation of serine metabolism. One key regulator is the mTORC1 signaling pathway, which integrates various environmental cues to control protein synthesis and metabolism, including serine synthesis. Additionally, transcriptional regulators such as c-Myc and p53 play significant roles in modulating serine metabolism in response to cellular stress and nutrient availability. The alteration of serine metabolism in various diseases, including cancer, neurodegenerative disorders, and metabolic syndromes, has sparked interest in its potential as a diagnostic or prognostic biomarker. Changes in serine levels or the expression of key enzymes involved in serine metabolism could provide valuable information about disease progression and response to treatment. Further research is needed to explore the clinical utility of serine as a biomarker in different disease contexts. Given the importance of serine in cellular processes, researchers have investigated the effects of serine supplementation on cellular metabolism. Studies have shown that serine supplementation can enhance cell growth, improve mitochondrial function, and influence various metabolic pathways. Moreover, serine supplementation has shown potential therapeutic benefits in certain metabolic disorders and may be explored as an adjuvant treatment for specific diseases in the future. Serine metabolism is intricately connected to cellular antioxidant defense systems. Serine-derived metabolites, such as glutathione and NADV, play a crucial role in combating oxidative stress and maintaining redox balance within cells. Glutathione, a tripeptide composed of glycine, cysteine, and glutamate, acts as a potent antioxidant, protecting cells from oxidative damage. NADV, generated through serine metabolism, serves as a reducing agent required for the regeneration of antioxidants and the detoxification of reactive oxygen species. Mitochondria, often referred to as the powerhouse of the cell, play a crucial role in cellular energy production, biosynthesis, and redox regulation. Serine metabolism has emerged as a key player in maintaining mitochondrial function. Serine-derived metabolites act as important cofactors and regulators of mitochondrial processes. The dysregulation of serine metabolism can impact mitochondrial function, leading to energy deficits, oxidative stress, and impaired cellular function. In conclusion, serine metabolism is a fascinating field of study that encompasses various aspects of cellular biochemistry, physiology, and disease. Understanding the intricate pathways and connections associated with serine metabolism provides valuable insights into the molecular basis of numerous diseases and opens avenues for potential therapeutic interventions. Through our exploration of the biosynthetic pathway of serine, the significance of SHMT, the interplay with 1-carbon metabolism, and its various roles in neurotransmitter synthesis, cancer biology, and cellular function, we have gained a deeper appreciation for the importance of serine in maintaining cellular homeostasis in human health. As future healthcare professionals, it is crucial to grasp the complexities of serine metabolism to better comprehend disease mechanisms and develop innovative strategies for diagnosis and treatment.